Hello, today I will be talking about the Golden Gate Assembly. So a little bit about how it works and what it is. This is a method used for assembling multiple DNA fragments. The Golden Gate Assembly relies on the usage of type 2 restriction enzymes, either BSAL or BBSL. These enzymes cleave outside the recognition sequence, resulting in 4 overhangs. So just a little bit about the steps. In Golden Gate Assembly, the two different types of restriction enzymes are designed within the gene of interest. These are used to create the 4 base overhangs, which I was talking about. The 4 overhangs are then used to assemble the DNA fragments. They are freely designed and termed fusion sites in Golden Gate Assembly, and then the fusion sites enable base pairing assembly of genetic parts such as the promoter, the coding sequence, and transcription terminator. So overall, by using this simultaneous restriction and ligation in one reaction, it results in a rapid assembly of multiple DNA fragments. In the final product, the type 2 restriction sites are absent, and the recognition site does not appear in the final product as well. So it's a precise and very clean cloning. So how is this related to some of the modules that we have been talking about? The Golden Gate Assembly method is very well ties into what we've been talking about in just cloning. Golden Gate is used to clone, to assemble multiple DNA fragments into a single piece. Similarly, we talked about the Gibson assembly, which is also what we've been using in our recent projects. The only difference is that Golden Gate assembly relies on the restriction enzyme and Gibson assembly does not. Some of the advantages around Golden Gate assembly, it can be done in a 30-minute reaction, so it's pretty quick, pretty precise, very good. The final ligation product is irreversible. This is due to the fact that the recognition sites are not included in the final product, like I mentioned before. It is high scalability, so what this means you can use up to 10 fragments in a single reaction. This is also due to the 4 base overhangs that you're able to input multiple fragments at a single time. In comparison with Gibson requires 20 to 40 base pairs of homology at the ends of the DNA fragments. And with gateway cloning, it produces a recombinant scar. As for Golden Gate assembly, which the end product is absolutely scarless. Some disadvantages to Golden Gate assembly is that it does not work well with vectors that contain multiple restriction sites. It has to be a single restriction site. DNA molecules that contain multiple restriction sites can be digested multiple times and get lost during the assembly. So that is why it only wants one restriction site. When using the type 2 restriction sites, it cannot be present in the fragments that are being used. So the solution to this is that you have to domesticate the fragment with PCR-based amplification before even starting the Golden Gate assembly, or the final product will not be what you want. It won't work, basically. So some of the controls that are required to do a Golden Gate assembly, you have the T4 DNA ligase, one of the type 2 restriction enzymes, an insert, vector, and PCR primer. Here's one example of how Golden Gate assembly is being used. So in this specific example, researchers were investigating gene function in Bacillus anthracis. They did cloning within protein and EAI allelic exchange vector. So they cloned it successfully. In this specific example, an internal, the B-cell I site was used as the restriction enzyme. And the plasmid was destroyed to avoid the unwanted cleavage. The DNA fragments were cloned between E. coli and BAM-Hi. So that can be seen on this first image that I have here on the side, is where they the DNA fragments were cloned in. And then on this second image, I have the rest of the cloning and ligation and the Gibson assembly with the final product. In this second example of usage of Golden Gate assembly, researchers were looking at plant vectors and tobacco plants specifically. So both the Golden Gate assembly and Gateway assembly were used. The Golden Gate assembly was used to assemble multiple DNA fragments in a predefined order. So in this first image I have here to the side was all the 4 vectors that were used during their experiment. And it shows on the notes section, you can see which areas were integrated with the type 2 restriction enzyme, either the BSL1 or the BSB1. And since Golden Gate can only arrange DNA into compatible vectors, not including the majority of binary vectors for plants specifically, which is what the researchers were looking at, that is why they continued on with the Gateway method. In this following image with that same research, you can see how the different vectors used were all compatible with Golden Gate assembly. So in order to test if they will all work well with the DNA assembly, they used Golden Gate assembly as a recipient vector to express fluorescent in the protoplast. So in this figure A down here, you can see how they use GFP, YFP, and INF1. They were all fluorescents, that the DNA were expressed, the vectors were expressed with the fluorescence to see if the DNA was assembled correctly. In this final example, researchers were using Golden Gate assembly for the host Pichia pastoris, which is a yeast. It has been found that Golden Gate assembly is the best method to optimize expression of the single recombinant gene for the production of this specific yeast. So what they did was the DNA products were integrated into BB1 and BSL, which is the restriction enzyme for Golden Gate assembly, and the fusion sites 1, 2, 3, and 4. So since this was designed specifically for the host, it was composed of 3 backbone levels. The first one was to harbor the 3 basic modules, the second one was to assemble transcription units, and the third one was to further combine multiple transcription units. So on the image to the right, you can see how it goes through the entire Golden Gate assembly from the PCR to doing the vector with the transcription enzyme into the Golden Gate assembly for this specific host. And these were just some of my other references.